Hi guys! I'm so sorry I'm not in school today, but I just wanted to give you a quick introduction to what we're doing today. So we're doing cell cycle CERs. So this is basically the same thing we would be doing if we were in the classroom. We'd be looking at cells under a microscope. So this is microscopic images of cells. So that, and we're going to identify what phase of the cell cycle each is in. So I just wanted to talk to you quickly about these. So when we look under a microscope, we can usually see a lot of cells on a slide. So each of these like little boxes is a different cell. Most of them are in interphase. So interphase is when we have a circular nucleus. You can see the nucleolus in here. So just like a dark circle in the middle, right? Once you start to see I call them fingers, right? Once you start to see the chromosomes, which look like fingers, that's when the cell is in one of the phases of mitosis. So I have the different phases here. So again, interphase is when it's just a dark mass in the middle. Once those chromosomes have condensed, now they look like fingers, okay? So prophase, the fingers are still together, but they're visible. So we can see the individual chromosomes a little bit more. They might be starting to spread out, not quite in a circle anymore, because that membrane is disappearing. But this is prophase. Metaphase is when they line up across the middle. So they're going to look like fingers that are still lined up. They're still together. Anaphase is when those chromosomes get pulled apart. The sister chromatids are being pulled to opposite sides of the cell. So that's what it would look like here. Okay, also over here, you can see them. We can see two separate groups of fingers, whereas metaphase, the fingers are together. I keep calling them fingers. That's how they look to me. And then telophase is when those fingers close into fists, because now they're starting to make new nuclear membranes, and we end up with two new nuclei at the end. Okay, so let's do the first one together and ignore this part because I think you just really can't draw arrows on a Google Doc. So whatever, that's for if we were in person. So we've got a cell here, and since this is a nice dark circle, it's clearly circular, so that membrane is still intact. We can't really see the fingers yet. So this is interphase, okay? The evidence is that the DNA hasn't formed into chromosomes yet, so we don't see visible chromosomes, and the nuclear membrane is still clearly visible. We still have a nice crisp border around the nucleus. So evidence, when you're talking about evidence, I want evidence from the picture. So what in the picture is showing you that this is interphase? Reasoning is where you describe from your notes, so make sure you have your notes out, what's happening during this phase. Okay, so describe using your notes what's happening in interphase. So in interphase, the cell is growing, it's functioning normally, replicating DNA, and getting ready to reproduce. Okay? So you're going to do this, and again, ignore this part where it says draw the arrows. You're going to do this for all of these examples. And so for each of these, just focus on the one cell that the arrow is pointing to. And some of them like this one, so station 1, it's not super crisp and clear. And that's on purpose, right? When we're looking in a microscope, it's not always super crisp and clear. So I just want to know, how does this cell look different from all the ones around it? So compare it to all these other ones. What phase is this guy in? Use evidence from the physical appearance to help you describe, and then use reasoning from your notes to explain what's happening in that phase. Okay? Same thing for each of these. Some of these are a little more clear images. Okay, if you have questions, please email me and I'm happy to help. Good luck.